people will start joining. So I wanted to welcome everyone. It's Friday, it's 7:30. We have yet another fantastic speaker. Kapil convinced me that he is a fantastic scientist when he got the first award of PhD in Germany, in Frankfurt, in Goethe University. And that is a pretty high honor. I'm sure the class was extremely competitive and he did it. And now we have this great interest as a field in stem cells and transplantation and a little bit of combination of all. So we're really delighted to have one of the leaders in the field. And Kapil, now I will pass it to Cesare, who is going to introduce you formally.
Thank you. Good morning, everyone. Can everyone hear me?
Yes.
And my name is Cezary Ritz, and I'm a postdoctoral researcher at Dorota Skowronska-Krawczyk Laboratory at Kevin Herbert Eye Institute. It is my great honor to welcome you to today's lecture and to present today's speaker, Dr. Kapil Bharti. Dr. Bharti earned his bachelor degree in biophysics in 1996 from the Punjab University in Chandigarh, India, where he graduated with the IS honors. In 1998, he obtained a master's degree in biotechnology at Maharaja Sayaji Rao University in Baroda, India. Dr. Kapil Bharti then moved to Germany, where he received diploma in molecular biology at the Johann Wolfgang Goethe University in Frankfurt, Germany. This was followed by a PhD in molecular biology at the same institution, which he obtained in 2003, graduating summa cum laude. His PhD work involved basic biology in the areas of heat stress, cellular chaperones, and epigenetics. After completing his PhD, Dr. Bharti moved to the United States and joined National Institute of Neurological Disorders and Stroke in Bethesda, Maryland, where he worked with Dr. Heinz Arnheiter as a postdoctoral fellow from 2004 to 2008. His time there resulted in numerous papers in the areas of transcription factor regulation, pigment cell biology, and the developmental biology of the eye. Dr. Bharti has authored numerous publications and has won several awards, including being a finalist in the prestigious Trans-NIH Earl Statman Symposium. Dr. Kapil Bharti currently serves as a senior investigator at Ocular and Stem Cell Translational Research Section at the National Eye Institute. Dr. Bharti's current research goals are focused on retinal pigment epithelium. The aim of his current research is to perform translational research on degenerative eye diseases. Without further ado, please welcome Dr. Kapil Bharti.
Thank you, Cesare, for a kind introduction, and thank you, Chris and Gazania, for inviting me. Do you see my full screen and do you hear me well?
Yes.
Yeah, wonderful. All right, so what I thought I'll do is I'll give you an overview of the kind of things we're interested in around, mostly around human RPE biology. And I don't need to explain this slide to this audience, but I wanted to put it here to kind of give you a sense of what our interests are. And, it really starts with this RPE being strategically located, evolutionary selected, sandwiched between the photoreceptors and the choroidal blood supply. And it's there for a reason that throughout its life, all this tissue does is maintains health and integrity of photoreceptors on its apical side and of the choroid on its basal side. And this tissue is able to do that because of its highly polarized nature. And what I mean by that is it has this very unique apical structures called apical processes or microvilli that interact with the outer segments of photoreceptors. I often call them the business end of RP cells. This is how RP cells interact with the photoreceptors. And on the basal side, it has again a very unique structure called Brooks membrane, highly proteinaceous, and allows free flow of macromolecules with the choroidal blood supply. This tissue is able to attain this polarized nature developmentally by the formation of this very specialized junctions between the two cells called the tight junctions, which are formed by many different protein complexes. So my lab has been interested for a while in kind of understanding how this tissue develops, how it polarizes, and how it attains all its functions. How does it interact with the photoreceptors on one side and choroid on the other side, and during in a normal healthy condition, but also how does that derail, and when this tissue is not able to perform this function, how does that lead to a disease outcome? And just to give you an example here, two diseases that I'm going to talk about today is At the bottom left, you see a case of AMD, an advanced stage of dry AMD here. We're interested in looking at this end here. How does RP dysfunction lead to sub-RP deposits? And with time, RP cells atrophy, and that leads to photoreceptor degeneration on one end, but also capillary loss on the other end. So we want to create models to study that phenomena as how do AMD disease processes spread across this whole tissue, starting perhaps in one tissue, maybe RP, maybe choroid is still a conflict there. Then on the other side, another example that I'll bring up today is another case of advanced AMD called wet AMD, where, as you all know, that choroidal blood vessels start to hyperproliferate and often grow what I call in the Z direction, where they may leak fluid or spread under the RPE and causing, again, in this case, photoreceptor degeneration through separation of photoreceptors from the RPE. So we're interested in how does RP lead to, or dysfunction of RP leads to wet AMD in one case or dry AMD in the other case. So for me, all of this starts with the model that we're interested in is iPS cells, induced pluripotent stem cells. Again, something that I don't need to explain to this audience. The beauty of this technology is that you can make any cell type of the body, but also you can start with human tissue. So you can start with healthy tissue, healthy donor cells, or patient cells. So you can connect anything that you're doing in a dish with patient genetics, but also you're doing things in a dish allows you to do very detailed molecular biology and cell biology of the tissue. We are now currently making in the lab RP cells that I'll show, cells of the choroid, and photoreceptors in collaboration with David Cam, the results that work I'm not going to show today. So in the first part of my talk, I'll focus on how do we actually make what we call functionally validated RPE cells. And during that process, like I said, we're interested in studying how do these cells become RPE, how do they become polarized, how do they become functional. So I'll show that in the first part of my talk. Then in the second and third part of my talk, I'll move on to showing some disease models that we have been able to create mostly around AMD. I'll skip the monogenic part today. That's perhaps for another discussion, but I wanted to keep it centered on one topic today, focusing on making use of these RP cells for making 2D and 3D models of AMD. Again, we are also very interested in, as Chris said, in developing cell therapies for many diseases, including AMD. One of our work with autologous RP patch for AMD patients is currently in phase 1 trial. I'll skip that part again for today's discussion, so focus on the left part of this slide. So first thing for me is, comes is how do you make RP cells from iPS cells, right? So this is my postdoc was in developmental biology of the RP. So for me, this is all developmental biology in a dish, and really trying to recreate what we have learned for many, many decades in animal models in other systems to recreate that in a dish to make human RP cells. So we spent quite some time focused on this top part, going from iPS cells and making sure the cells, as you go towards RPE, would follow the various developmental stages, neuroectoderm, RP progenitors, committed RP, all the way to mature RP. And again, this is combining knowledge and work of many, many labs and some of the work that I did in my postdoc to add the right growth factors at the right time points and looking for right biomarkers, mostly transcription factors that they're coming up at the right time for the cells to become RP cells. This has several advantages of following this procedure is that you make sure that the cells have the right epigenetic marks of the right genes and the right promoters for them to become more native-like RP cells. The process becomes more efficient, more reproducible. And as you can see at the bottom here, bottom left side, you see a dish full of pigmented RP cells. In the middle, you can see fully using a flow marker, highly pure RP population, over 98, 99% RP cells, we can consistently obtain this population. And in terms of reproducibility, I can show you that we have now done it over 50 different donor iPS cell lines, over 100 different clones of iPSC, and over 500 different differentiation runs, and many, many different disease conditions. We have shared this protocol across 24 labs, half a dozen companies, and we're quite happy that many, many labs have been able to reproduce this protocol. We still continue to be interested in understanding how this process happens. And actually I put this up here because I wanted to tell you that we're now trying to actually understand how does differentiation happen in 3D. So we're trying to 3-dimensional imaging of the cells as they're maturing into RPE and combining that with AI and image analysis to really recreate RP differentiation in 3D. But I want to just go back to a few years when we were optimizing this protocol. One of the things that at the time in the stem cell field overall was a big problem was that people could make many different cell types from iPS cells, but always the issue was that the cells were locked in some kind of an immature fetal-like state. And since we were interested in recreating adult phenotype in these cells and modeling adult diseases in the cells, we wanted to make sure that we pay extra attention to making these cells, converting these cells into fully mature functional RPE cells. So how did that come about for us was, is that while we were understanding where the cells are maturing or polarizing, one of the feature, of course, of polarization of these cells, as I said, is apical processes. On the left, you see a scanning EM of an RPE surface where the apical processes have not yet quite come up. They're starting to come up from the sides. But what we noticed was that preceding the apical process formation was an interesting structure in every cell that was coming up, which is this tall structure called the primary celium. I'm sure most of you are familiar with this. This primary celium is a cellular appendage, a subcellular organelle, which, as far as I understand, is present in at least at a given stage of at least every somatic cell type. And this structure is formed by 9 triplets of microtubules that push out the cell membrane and stick out of the membrane. And now over a lot of work has been done on this in understanding this work, in understanding the role of this structure in cells. And it's a lot of this suggests that it is an important intracellular signaling hub and regulates many different signaling pathways, including planar cell polarity, cytoskeletal organization, cell cycle, and many different aspects of the cell. And of course, also something that I'm sure all of you are familiar with, that defects in primary celium are now associated with a very large class of diseases called celiopathies. This table shows kind of overview of some of these diseases. When I started working on this, I collaborated with a very talented colleague of mine, Helen Nisamera, who is an expert on primary celium, and now she runs her own group in University of Mainz in Germany, still working on primary celium and RP biology. So what we were able to put together is that, of course, again, published work from others has shown that many different syndromes are associated with celiopathies or associated with mutations or defects in primary cilia. And retinopathy was a very unique, very common phenomena in many of those cases. We focused on 2 different diseases, Zubair syndrome and LCA, which were caused by a mutation in a protein called CEP290, which is a basal body centrosome component and is involved in mediating ciliary protein trafficking. And another protein we focus on was BBS8, which is an adapter protein for ciliary trafficking and a component of so-called the BBSome, and its a mutation in this protein are associated with severe planar cell polarity defects. So first thing what we did was, of course, what our model is, made iPS RPE cells from one of the patients with CEP290 mutation. And this work led by Helen and in my lab done by Chin and Ruchi Sharma showed that, first of all, what you see on this is on top left is healthy iPS cells expressing ARL13B, which is one of the markers that stains the exoneme of the primary celium. CEP290 is present in the basal body under the celium in healthy cells, and obviously in patient cells where there's mutation in CEP290, it's missing in those patient RPE cells. But when we looked at the cells carefully, we realized that the primary celium in the patient cells were significantly shorter than the healthy RPE cells. And when we started analyzing the phenotype in the cells, we noticed that the apical processes of the patient with CEP290 mutation, they were messed up. So when I say messed up is, if you see on the left side, you can see very dense, nice packaging of apical processes. On the right, what you see is weird balloon-shaped apical processes formed in the cells, as if there was a misfiring of protein complexes or vesicles that make the apical processes, and there was no control left how the apical processes are formed. So we're quite intrigued by this observation that a defect in primary celium is somehow associated with a defect in polarization of RPE cells. So we looked deeper, and notice that these cells were also not fully maturing completely. So for instance, there was, when we looked at expression of about 70 published adult RP specific genes, this is work done by Sheldon Miller at NEI, and we saw that patients, up to 90 patient RP cells had about 67% of those genes were downregulated, adult RP specific genes were downregulated, as if these cells were not fully maturing. And of course, functionally also, we saw that these cells had reduced ability to phagocytose outer segments. What you see on the right side here, green are the CEP290 cells. The ability to phagocytose was almost half, less than half of the adult RP cells. So this work is published, but I'm going to summarize it here briefly. So what we ended up showing was that what is happening is that BBS8, which is also localized at the centrosome of primary cilium, it interacts with two different proteins and recruits them to the primary cilium. One of them is zuberin. Zuberin is a transcriptional co-activator of beta-catenin, which is a canonical Wnt signaling activator. And when BBS8 recruits zuberin to primary cilium, it turns off the transcription activity of beta-catenin. And the second protein, inversin, as it recruits it into the primary cilium also, it leads to degradation of the scaffold, which leads to stabilization of beta-catenin destruction complex, leads to degradation of most of the cytoplasmic beta-catenin. At the same time, we were able to show that the primary cilium activation or formation led through calcium signaling, activation of PKC delta and Rho kinases, which led to cytoskeleton reorganization and solidifying of the cytoskeleton around the cell borders, tightening the tight junctions, also causing leftover nuclear beta-catenin to move there, forming cells, making cells fully polarized. So this work then led to this hypothesis that if we were to activate formation of primary cilium in RPE cells, in iPS-derived RP cells at a given immature stage, we should be able to synchronously polarize the entire monolayer in our culture system. And in fact, that's what we tested and we saw. So to do that, remember that timeline I show of making RP cells from iPS cells. At the immature RP stage, we add either of the two different chemicals, acetylcholine or PG2. Both of them have been published and known to induce celiogenesis. We saw that both chemicals were able to induce celiogenesis in our cells as well. And again, you can see individual cilia labeled with the two different cilia marker, ARL13B and GT335. So after we confirmed that these chemicals were able to induce celiogenesis in our cells, we checked whether the cells were polarizing or maturing better. Here's an example, for instance, if the cells would mature, beta-catenin, the transcription factor that normally is in the nucleus making RP cells as they develop, should move out of the nucleus and align itself around the membrane. And this is exactly what we see here. You can see beautiful red staining all around the cell membrane. Beta-catenin, it's kind of parsing itself and moving out of the nucleus, giving us a good hint that these cells are indeed maturing when we activate primary cilium in these cells. Chin and Rucci were also able to do transmission EM. This is a side view of a cell, again, looking at whether cilia induction, for instance, in this case with acetylcholine, similar results with PG2, was able to change structural phenotype of the cells. And here what you see is, as compared to non-treated cells, we see massive upregulation in formation of apical processes, very dense network of apical processes, significantly increased pigmentation in these cells. And that is also directly translated, as Chin would show, using electrophysiology assays, and these cells become very similar in their phenotype to native RP cells in their electrophysiological readout. So what you see here is several features of the cells. First, on the right side, you see transepithelial resistance. This is a feature that is directly coming from how mature the tight junctions are between neighboring cells. As the tight junctions form, if you put an electrode one on top and bottom and you pass current, cells will show resistance to the flow of current. And we know that increase in resistance is almost directly proportional to how mature the tight junctions are and how mature the cells are. And as the cells become polarized and the tight junctions become so tight that they do not allow the channels and the receptors on the apical side to exchange with channels and receptors on the basal side. So cells would have different channels on one side than on the other. That would lead to differential membrane potential, which is what we can see here on the left side here. These cells have about 4.5 millivolt transepithelial potential, very similar to native RP cells. And then we can do physiological perturbations in the system, for instance, lowering the potassium concentration in the apical bath from 5 to 1 millimolar, something that not naturally happens in the eye. Every time light hits the photoreceptors, the potassium channels shut down, potassium concentration drops in the subretinal space. RP cells hyperpolarize, open up their potassium channels to correct the subretinal potassium concentration so that photoreceptors can perceive light for the next photon. And that's exactly what we see in the cells. Another assay we did is looking at when light hits, it's thought that photoreceptors secrete ATP, which binds to cholinergic receptors on the RP apical surface, causing activation of calcium stores, intracellular calcium stores, which leads to opening of basolateral chloride channels. So we see classical biphasic depolarization pulse from these cells. Again, very similar features to native RP. So all of these assays give us huge confidence that what we're making in vitro is very similar to native RPE cells. But we kept digging deeper as we kept making these cells and kept looking at them more. We got more and more interested in their structural features and what different types of RP cells we're making. And that brought us to this beautiful paper published almost 50 years ago by Roy Sternberg and colleagues from UCSF, who were looking at, in this case, actually cat RPE cells and analyzing their structural features. What they noticed was what's summarized on the left side here with hand drawings, I think, is that there were two different types of apical processes on RPE cells. As all of you know, that rod outer segments are much taller than the cone outer segments, so rod outer segments can go deeper into an RP cell. So RP cells have to have very small apical processes that can interact with the outer segments, whereas for cone, the outer segments are shorter, so RP cells send out their apical processes all the way up. And these apical processes, as is shown here on the right side of this image, is they are like sheets of cone. They wrap themselves around the cone outer segment. So they're very distinct two different types of apical processes, which is also now illustrated here by the scanning EM. Where we can show what we call finger-like apical processes, which would interact with rod outer segments, and paddle-like apical processes, which would interact with cone-like outer segments. At the same time, groups have shown that, so you can imagine that this kind of RP cells would be more enriched in the macula, which is cone enriched, and these would be more enriched towards the far periphery, which is rod enriched. And at the same time, many groups have shown that cells in the macula, RP cells in the macula, are much smaller in size as compared to RP cells in the far periphery, which are larger in size. So we were curious to understand what kind of RP cells are we making when we make RP cells from human iPS cells. So to do that, we decided to set, first of all, a benchmark for us. And Davide, with Ruchi and Arvidas's help, kind of decided to do it in a very systematic way. The first thing they did is develop an artificial intelligence-based imaging network to understand, to image and segment human RPE cells from a human eye flat mount. So what this program does is you just stain the RP borders and we image an entire human eye. You talk about almost 80 different images that are then stacked together. And then the artificial intelligence-based network, what it does, it segments the borders of each cell and then analyzes its shape parameters. It analyzes about 25 different shape parameters and then assigns different color based on heat map, how small or big the cell is. For instance, here the smallest cell will be dark blue, and as the color changes towards light green or red, the cells are becoming bigger. So here's an example of a complete map of a human eye, single cell resolution map. In this map, 3.5 million cells have been analyzed. So what you see is, I'm showing it to you in such a low resolution. Each dot is actually a single cell on this map. But it has, as soon as we saw this map, we were fascinated with this because it has several beautiful features to it. First of all, you can quickly recognize the macula here in the center, which is the smallest cell. So we know that what we're doing is correct. But then it has, what you see is, as you go outwards, it has 5 eccentric rings of different type of RP cells, different sizes of RP cells. So macula has a small, then there's a ring of large RP cells, and then there's a eccentric ring of intermediate, small, large RP cells, then large again. And then there's a very interesting new ring all around the eye of RP cells that are in size, very similar to macular RPE cells. So we're really intrigued that what's going on here and whether this map is reproducible across different species. So there's we call them now 5 different populations: P1, P2, P3, P4, and 5. Now this will become useful later as we look at eyes from different diseases. So now, so first thing we were intrigued is whether this map is unique to each species and how does that look and how does it come about, right? So what Davide did and started now working on with help with Dominic, who's a PhD student just started in my lab, is started making this map across many different mammalian species. So here's an example on top, which you just saw, a map from human eye, map from a monkey eye, which is again has we can recognize the macula, but the map is quite distinct than human eye. And mind it, in each case, we're looking at least a dozen different eyes, so we know the map is consistent across different eyes in one given species. But for instance, in monkey, the map correlates with the known cone density in monkey eye. For instance, of course, the highest is cone density in monkeys, as in human is in the macula. But then second, it stays high around the macula, and then it drops quickly where we see larger cells. So again, every time we see small RPE, we see high cone density correspondingly. We work a lot with the pig model for our cell transplantation work, and so we had access to pig eyes. And in pigs, we do this work in what is called the visual streak, which is equivalent of macula in human and monkey, where it is well known that there's a high cone density and goes from kind of a nasal to temporal, or temporal to nasal end of the eye. And again, we see smaller RP cells in that area. If we look at rodents, mouse and rat, which are not known to have macula or visual streak or high acuity area, we don't see similar pattern. We don't see any dense small RPE cells. In fact, it is known that in mouse, there's a high density of cones towards temporal inferior part. And that's where we also see randomly speckled small RPE cells in between large RPE cells. So we think that really RPE cell size really correlates with the density of cones in different species, and really it opens, immediate question comes to your mind is, well, so what comes first? Is RP map instructive to photoreceptor differentiation, or is the photoreceptor map instructive to RP differentiation? And Dominic is trying to address that question. But with that, going back to what we were interested in, kind of the translational aspect of this. So we looked at some of the eyes from AMD patients, and the map was like, looked like devastated map that it didn't work at all. But what happens here is the entire P1, the macula is gone. These are really late stage AMD eyes coming from cadaver eyes, obviously. But interestingly, the P5, what we call P5, which is, remember, in size is almost identical to macula, is also gone in these eyes. So we think there's some kind of a vulnerability of smaller RP cells in general for diseases like AMD. But then what happens in other different diseases? So what Davide did together with their clinical colleague, Larissa Hearn, in our NEI clinic, they put together a table by analyzing wide field fundus photographs from many different patients with different diseases. For instance, here we have samples from eyes fundus photograph from choroidemia patients, where by doing measurements, because from our map we have exact millimeter measurements of different RP populations. So when we transfer those measurements to those wide field fundus photographs, we can say, we can tell precisely that in choroidemia patients, the damage is dominantly in P3. In LOD patients, damage is in P2 and P3. There's an undiagnosed retinal degeneration patient we have where the damage is in P3 and 4. Stargardt's dominantly in P1, and AMD P1, P4, and P5, like I just showed you. So that immediately again opens another very interesting question: is, what are the molecular and functional differences in these different RP populations that dictate their differential sensitivity in these different diseases? So without, how do we address this kind of question, right? And we, as I said, we are interested in making RP cells from iPS cells, and we think we're good at making them. We thought, why don't we try and make all these different populations, so then we can study them in vitro and see what are the differences in those populations at molecular level, and how they're differentially sensitive to different diseases. So that's kind of an ongoing work in the lab. I'll quickly give you a spiel of this. What Davide and Ruchi have been able to do is they use the same artificial intelligence network, but then while they were, and this was now done as RP cells were differentiating from iPS cells, and at the immature stage, they added about 110 different compounds, which would upregulate or downregulate various different developmental pathways. So we made a list of all the known developmental pathways because we thought it making different type of RP cells, it's got to be a developmental phenomena, right? So there has to be some developmental pathway that is responsible for doing this. So we made a list of about these 110 compounds, and we added them one by one at this stage. And we asked our network to say, sort out cells, compounds that make smaller cells, and sort of compounds that make larger cells. And then we'll figure out if those cells are indeed peripheral or macular cells. And indeed, that's what happened. So we were able to pin down a few compounds that can make pure macula-specific RP cells and what we call pure mid-periphery specific RP cells. So what I'm showing you here in scanning EM on the left is apical processes that are all those pedal-like apical processes. Every cell has those, whereas on the right side, I'm showing you cells that have mostly finger-like apical processes. And I hope you can also appreciate difference in size of these cells. These cells are bigger, these cells are smaller in size. We just ran through a single cell RNA-seq analysis of this data. And again, we can, these cells are all RPE cells, right, for the bulk gene expression, but we can still sort them into different subpopulations. Here on top, you have in blue is macular RP cells. In the middle is a perimacular, what we used to make before, P1 to P2 population. And then we have a P4 mid-peripheral RP population that we can make. So now we are now still, as this is ongoing work, we're still trying to understand more what are the physiological and metabolic differences between these different populations. And then we'll translate that into their differential disease sensitivity. So with that, I hope I've convinced you that we have spent quite some time making and understanding RP cells, human RP cells that we make from iPS cells. I want to transition to the utility for two different disease models. So I'm going to first start with dry AMD. We were interested in understanding how RP cells dysfunction would lead to formation of sub-RP deposits, and that, of course, is thought to lead to RP atrophy with time and their cell death. So again, I don't need to tell this audience that AMD is a disease, is a complex disease. There's a lot of work done using GWAS studies that has linked several genetic factors to AMD. Epidemiological studies have linked diet, smoking, and aging to AMD. And it is thought that all of the combination of these two leads to some kind of a chronic stress in the photoreceptor RP chloride interface, leading to inflammation. There's a lot of thinking around this abnormal complement activation, which is thought to lead to RP dysfunction, drusen deposits, eventually to advanced AMD. So we will focus on the chronic inflammation abnormal complement path, and more specifically, we'll focus on alternate complement pathway. And the reason for that is, for those of you who are not familiar with complement pathway, there are three different arms of complement: classical, alternate, lectin. All three of them eventually can lead to the same membrane attack complex. Classical and lectin are more classically known to be activated by microbial pathogens, whereas the alternate complement can also be activated by several intracellular non-pathogen related, intra-body non-pathogen related processes, and it can also be activated by slow hydrolysis, what is called the takeover mechanism. The way this works is interaction between complement factor B and D activates B, which binds to complement C3, activates what is called a complement C3 convertase and breaks it down into C3b and C3a, and leads to formation of a C5 convertase, breaks it down to C5b and C5a. These B peptides or proteins go on towards making a membrane attack complex for the downstream complement proteins. But interesting for this talk are C3a and C5a, the two peptides that are called anaphylatoxins, which in the immune system have been known for a long time for their role in regulating inflammation, chemotaxis, muscle contraction, even autophagy. And they do this via direct binding to C3a, C5AR1 receptors, both on the membrane surface and actually intracellularly as well. So our interest in alternate complement pathway was through what is marked here with asterisks, is because several proteins in this pathway have been linked to AMD. They contain risk alleles for AMD. CFI and CFH, both of those are inhibitors of the pathway and have risk alleles for AMD. CFD, B, and C3 have all risk alleles for AMD. So what we thought, what if we check the activity of alternate complement pathway on iPS-derived RP cells, what do you think we will get? And this experiment, that the next part I'm going to show, is the work done by 4 people, predominantly 4 people in my group: Ruchi Sharma, Malika, Devika Bose, and Davide. And what they asked was a very simple question, that if we add activated complement onto RP cells, and there's some precedence for that in literature from work published by Link Johnson and Kristen Curcio, that RP cells in culture can make drusen-like deposits. So what happens is, what you're looking on the left side is a kind of a transverse view of an RP monolayer grown on a transfer filter that we normally use to grow them. In red is ApoE, affectin labels the borders. ApoE is a known marker for drusen, as you all know. And normally, healthy cells have ApoE predominantly apically located. When we add the activated complement to RP cells within 48 hours, so it's a very acute stress, we see ApoE moving towards the basal side, secreted by RP cells. We can see all the pores of our transfer filters are clogged with ApoE. Cells look completely messed up, as you can see, affecting staining. So the cells start to undergo degeneration. We've characterized these complement or these secreted proteins in complexes in many different ways. And here what you see is H&E staining of RP cells treated with the complement. You can see this massive buildup underneath RP cells that even kind of push away the monolayer and break it into pieces. And the bottom you see oil droplets stained with different dyes here, and you can see intracellular and extracellular oil deposits. So we think that these complexes have both lipids and proteins, and obviously ApoE as a lipoprotein is there. So what is causing the formation of these complement sub-RP deposits by activated complement? So first of all, we looked at whether RP cells do express C3AR and C5AR1 receptors, and indeed that's the case. And interestingly, these are present dominantly on the apical surface of RP. We correlated these results with cadaver human eyes, where we see these receptors both on the apical and basal. But in our cells, we see them dominantly apical. We see some basal expression, but it's dominantly apical. Now, to test whether it is the activity of these C3 and C5 through this receptor that is causing this defect in RP cells, we did what we call depletion experiment. So simple, we took sera that were depleted in C3, complement protein C3 or C5, or used different chemicals to directly block the activity of complement receptors. So what you see on this graph is we're looking at ApoE deposits as compared to inactive complement on the left, active on the top. We see increase in ApoE deposits, and that increase can be reduced by either depleting C3, C5, or by directly blocking the 2 receptors. Same is true for the degeneration of RP cells, the transepithelial resistance that we use as a measure whether the cells are degenerating. Is here we see that again, inactive complement compared to active complement, transepithelial resistance drops significantly, but in neither 3 cases where we have depletion of C3, C5, or we block the receptors, we don't see drop in transepithelial resistance. So this really gives us confidence that what we see in our system is indeed going through these receptors. So then, to understand what are the intracellular pathways that are being involved in this phenotype, we saw that active complement was increasing the activity of AKT and ERK kinases. Both phosphorylation of both of these kinases was significantly increased in these cells. And then downstream of this is known as one of the pathways is NF-kappa B activation, which we saw that in the inactive complement with strong increase in P65 translocation to the nucleus, suggesting that NF-kappa B pathway was activated, and downstream target genes for NF-kappa B were significantly increased. Here's an example I'm showing of IL-18, both on the apical side and on the basal side. We see increase in IL-18 in the presence of active complement. And we looked at other NF-kappa B target genes both by RNA-seq and targeted analysis, and we see strong upregulation of NF-kappa B downstream of active complement. At the same time, we also, just to prove that NF-kappa B indeed was upstream of ApoE deposits, we took advantage of another iPS cell line that comes from patients called patients with NEMO syndrome. So NEMO is an adapter protein that is known to negatively regulate the activity of NF-kappa B pathway. And in these patients with mutation in this adaptive protein NEMO, they're known to have much higher NF-kappa B activity. So basically constitutively active NF-kappa B. And that's what you see here when we received iPS cell line, made RP from patients with NEMO mutations or mutations in the protein NEMO. We see under normal condition, then the baseline condition, increased P65 nuclear translocation and increased upregulation of sub-RPE ApoE deposits as quantified here. We see much higher ApoE deposits, suggesting that NF-kappa B pathway is indeed upstream of ApoE formation in RPE cells. So without the presence of any complement, we could replicate this phenomena in these cells. At the same time, we saw as NF-kappa B activity was higher, we saw a complete downregulation of cellular autophagy phenomena. This has been known in immune cells, but we're happy to see that the similar things happen in RP cells, that in the presence of active complement. Here on the left side, you see ADG5, ADG7, LC3, all three different markers for autophagy. All three are downregulated in RP cells that are treated with active complement. Western blot quantification is shown on the right side. It's exactly the same results that we see both by immunostaining and Western blot downregulation of autophagy in RP cells. So then to kind of put this whole project together, what our working hypothesis is that there is a slow C3C5 formation, C3A, C5A, or C35 activation on the apical side of RP. The source of that, we think RP cells secrete. We have seen by ELISA that RP cells themselves secrete C3 and C5 proteins more so under stress condition, and it's been published by Bob and others group that RP cells do make C3, C5 higher under stress condition. Similarly, other groups have shown that photoreceptors can secrete C3-5 protein. So we think there's a slow grade C3, C5 activation happening on the RP apical surface, which perhaps is kept under check by CFH and other inhibitors that RP cells secrete, or also present on the RP surface of membrane receptors. But with time, as that takeover mechanism or that secretion of these proteins or this activation of these complement proteins goes beyond the threshold that these inhibitors are not able to keep them in check, they would bind to their cognate receptors through AKT, ARK, cause increased upregulation of NF-kappa B, downregulation of autophagy, leading to this massive sub-RP deposits, causing also degeneration and loss of tight junctions and overall stress in RP cells. So this working model immediately suggested an idea that if this is the case, then RPE cells with the CFH risk allele, one of the highest known risk alleles for AMD, should have a higher baseline deposits or should have higher disease sensitivity. And so we looked at that, and in fact, and indeed that happens to be the case. So here what you see is in this graph, we're measuring lipid deposits underneath RPE cells from two different cell types, CFH-YY, so 402YY allele, and CFH-402HH allele, both homozygous. And in this category, we have data summarized from 4 different lines, and here data is from 3 different lines. So it's not from one particular sample. And you can see many different technical replicates. What we see is that the white 402H allele already at baseline has higher lipid deposits. And then, of course, both would respond to complement because it's a strong acute stressor, but these respond stronger as compared to the YY allele. So we think really this complement, this model, I would say, provides a way to dissect more in detail how alternate complement pathway that is now known as a biomarker for AMD interacts with the genetics of the cells and does intracellular defects, causes intracellular defects in RP cells leading to disease pathogenesis. And this, to me, as projects has contributed as a proof of concept to what now NEI has started as a large ARIDS2 data bank of iPS cell lines from ARIDS2 patients in an initiative called AMD Integrated Biology Initiative. So overall, I think by now 64 different iPS cell lines with different risk alleles have been made. Some of the top lines, the top 10 lines, actually 11 lines are listed on this table. All of these lines have risk alleles in CFH, CFB and C3, but 6 of them are homozygous non-risk for the ARMS2 HTRA risk allele, and 5 of those are homozygous at, at the risk for the ARMS2 HTRA allele. So we think this really the two highest, so this would be the highest risk and this would be second highest risk lines, provides a very valuable resource for the community to be able to further dissect these pathways. And what another contribution that we have made is, as these cells were made by the New York Stem Cell Foundation, the agency that NEI contracted to make these cells and distribute to the community, we have actually validated that all of these iPS cell lines make RPE cells. So at least when going into these experiments, people have confidence that these cells will make RP cells in their hands, and this work is done by Roba, Devika, Omar, Kelsey, and Ruchi in my group. So, but one has to think of that AMD is not a disease, it's not a monogenic disease, it's not a straight answer that this mutation leads to this phenotype, and that's the mechanism. As we can see, the alternate pathway may be just one of the many different pathways we already know from work of many different people, oxidative stress pathway, mitochondria, lipid homeostasis in general. There are many different pathways involved in disease pathogenesis. So how do we really understand at a global level what AMD is and how is it caused? And I think this part of this initiative is kind of an artificial intelligence way to do in silico interdisciplinary research. So what I mean by that is what we hope to accomplish with all of this data is that for all of these iPS cell lines that I described, these 11 and the other 52 lines, we have patient data from EDITS 2 cohorts from in collaboration that we have obtained from Emily, and the genomic data again from Emily Chu at NEI. And Kerry Goetz is working on a database where all of this is being put together. And the goal is to now put the cellular data derived from iPS cell, derived RPE, photoreceptor, perhaps endothelial cells, whatever the interest of the community would be, to put it together into the same database and see if AI can find signals that we can't, because the data sets are too big and too complex for us to figure out. So I think that's perhaps one way of trying to make a sense of the 3 data sets for AMD. So this is where I'll again transition now to a 3D model, because I'm RP centric when I work, but I understand that disease doesn't happen in one cell type, doesn't happen in one tissue. Disease happens perhaps in multiple cell types, and its disease processes, how they spread across different tissues, is what eventually leads to final disease outcome. So really, we want to understand disease in its complete sense, and we want to replicate human disease in its complete sense in a dish. And for that, we want to create 3-dimensional models with using, again, iPS cell technology. And for that, I'll show you a couple of examples of what we're doing in the lab. But for that, first thing comes is how do you need now all the cell types for making those 3-dimensional models, right? So I already showed you that we can make RPE cells. And then now work that Roba and Tisun has done in my group, they have also been able to make endothelial cells, pericytes, and fibroblasts, taking iPS cells into a mesodermal fate. So now you have eventually the possibility actually of making patient-specific 3D tissue, so syngeneic 3D tissue with all the cell types in a tissue, for instance, RPE, and the cell types of the choroid in this case are all coming from one tissue. So what happens is, so once we make these cell types, so we can make RPE, we can make endothelial pericytes and fibroblasts, how do we put them together to make, let's say, RPE choroid 3-dimensional tissue? So for that, this was an effort that was started a few years back in my group by Minjae Song to 3D bioprint different cell types to make choroids. So Minjae is now running a bioprinting core at NCATS, and this work has been taken over by Russ Quinn and Eric Nguyen, PhD and postdoc fellow in my group. And what these people do is they put together these cell types in a hydrogel, and we call that bioink. And you can load that bioink into, this is what you see on top is a 3D bioprinter. You can load those bioinks in this print nozzles, and there are 2 print heads. Each one is a very fine syringe that can move in a precise XYZ orientation that you can control by a computer program, and then print any pattern you like. So what I'm showing you here is kind of a zigzag pattern that we make. And the reason why we're making that here is that the place where the bioink is deposited, that's where the cells are in highest concentration and gives them time to come together and start making capillary-like network. And those empty areas in between is where the cells would actually undergo angiogenesis and proliferate from one end to the other, and allows us to quantify how much angiogenesis is happening. So how does this technically work? Let me quickly check time. So we start with a biodegradable scaffold. We flip it on one side. We bioprint the bioink on one side of the scaffold, flip it upside down. We see RP cells on the other side, and we let this tissue mature for about 4 to 5 weeks. And during this time, the scaffold is gone, is degraded away. It's a biodegradable scaffold. Both RP cells and endothelial cells secrete ECM into this, what the space of scaffold would be, and replace that tissue with a Brooks membrane like structure. So I'm going to show you structurally and functionally with that. We have tried to validate this tissue as a complex RP choroid tissue. So first on top left here is on the scanning EM side view of the RP, Brooks membrane, and the choricapillary structure. So here's the RP cells. I'll show you more pictures of this actually at the bottom here. But what I want you to focus on is the structure in between RP cells and the choriocapillaries, the so-called our Brooks membrane. You can see here the thickness of this is about 2 microns, so exactly the size of a native Brooks membrane. And it really looks like how the Brooks membrane would be in a native tissue. When we stain it for different components of Brooks membrane, fibronectin, laminin, collagen 4, and elastin, we can see all of them, they are present in this structure. And actually, if we co-label them, we kind of see that they have a certain layering pattern in this Brooks membrane. By TEM, we followed that the tissue is maturing, RP cells are polarizing. At 3 weeks, we can see RP cells are not as pigmented, apical processes are there, but not as confluent. You can see capillaries formed at the bottom, there's Brooks membrane structure in between. And by 6 weeks, it's completely replaced by ECM, the scaffold is gone. The beauty is, and you can see tight junctions between neighboring cells, the apical processes, dense melanosomes. RP cells, when grown on plastic, do not form basal infoldings, but on this three-dimensional surface, we can see extensive basal infoldings, which is again a very critical feature of RP cells that is usually missing in cultures, but we have been able to recreate in this three-dimensional structure. This tissue is not only structurally looks like RP cells are mature, but also functionally it's mature. Again, we looked at transepithelial resistance, whether these cells have structural integrity. Here on the right side, you see our 3D, what we call outer blood retinal barrier, 3D OBRB, as compared to RP in 2D. The transepithelial resistance is almost similar in 2 cases and much higher than just the empty tissue by itself. So we know structurally this tissue is intact. When we looked at the bottom side, the choroid side, and you can see here endothelial cells are labeled in green, pericytes in red. You can see capillaries that run throughout the tissue. Some capillaries run from one end of the tissue here, you can see, to the other end of the tissue. But the beauty is the pericytes have wrapped themselves all around endothelial cells. You can also see here by transmission EM, pericytes wrapping themselves around the endothelial formed capillaries to stabilize them. And by lumen, they are a pattern of 10 to 20 micrometer lumen, very similar to native capillaries. So this is now looking at the tissue from top to bottom, so we can see both RPE and the capillaries at the same time. It's labeled in green for apical process marker ezerine and ecadrine marker red on top, and PLVAP and CD31 to mark endothelial cells, and PLVAP to mark actually fenestration in capillaries. So when I play this video, you'll see RP monolayer on top and a dense capillary network at the bottom. It comes back up. You can see again dense capillary network and RP cells on top. So what we do is, and then we do segmentation and image analysis of this tissue so that when we make a wet AMD model, we can see if the capillaries are at a different location. So what I mean by that is, here's the depth code. So if the capillaries are in the choroid, they are green. If they are going into the closer to the Brooks membrane or into the Brooks membrane, they may be yellow, and if they're above, they may be red. So as you can see in the wild type tissue, most of our capillaries are at the right place, a dense network, they're all green, and I'll show you later that how they change phenotype when we stress the RP. But the PLVAP expression suggested that these cells, our capillaries, may also be fenestrated. Fenestration, as many of you know, is a hallmark feature of choroidal capillaries, like a couple of other tissues around the body. that are small 50 to 100 nanometer pores in the membranes of endothelial cells that allow free flow of macromolecules from the blood circulation into the RPE. And you can see here on top fenestrations in our capillaries as compared to published work from Jerry Luddy's lab where they've kind of analyzed capillaries in fetal human choroid. So very similar pattern of fenestration is seen in both tissues. And interestingly, that this fenestration formation in the 3D tissue is actually directly dependent on the presence of RPE. What you see on the right side here is this top tissue contains both RPE and choroid, stained in red for the fenestration marker, green for endothelial marker. You can see nice angiogenesis happening throughout the tissue, and in higher magnification, you can see strong expression of fenestration marker and around endothelial cells. But in the absence of RPE, the tissue doesn't proliferate at all. There's no angiogenesis happening, and of course, there's also no expression of the fenestration marker. So really, as has been suggested in the past, RPE cells do regulate choroid angiogenesis and fenestration in our tissue as well. We went deeper into the analysis of the choroid by single cell RNA-seq here. So what you see on the top left is a TSNE plot where we're looking at in red are endothelial cells in 2D and in blue are endothelial cells in 3D. We noticed three different clusters of 3D endothelial cells, and when we looked at those gene expression more closely, there was one cluster that had high expression of mature choroidal markers, which are also shown here on the right side, KDR, CD34, PLVAP, PCAM. They all were present in this cluster. And then we saw one that was differentiated but not choroidal yet, and then there was an inflammatory cluster. So we're still figuring out why do we have three clusters of choroidal and of endothelial cells in this tissue, and we think it is based on the location in the tissue. So this is a side view on the bottom left, what you see. We think that the ones that is mature choroidal is the closest to RPE, and interaction with the RPE makes it into choroidal endothelial cells. The differentiated one may be deeper into it, and the inflammatory may be more deep into it. And since our tissue does not have perfusion at this point, it's probably increasing inflammation in those cells. But we're still trying to figure that out. So last couple of slides, I want to show you that we have also been able to functionally or translationally validate this tissue. We wanted to create a model, a disease model of this tissue and see whether it really works, for instance, creating a wet AMD model. And we chose wet AMD because it's so well characterized in terms of some of the pathways and how to block wet AMD phenotype. For instance, it's known that CNV is caused by higher VEGF secretion. And if you add anti-VEGF antibodies, you can block CNV formation. Upstream of VEGF, it is thought that HIF-1 alpha upregulation, perhaps induced by hypoxia, causes all of this. So we asked the question whether if we increase hypoxia in our RP cells, increase HIF-1 alpha expression, do we increase VEGF and then CNV in this tissue? So first of all, for inducing hypoxia, because we wanted to do hypoxia only to the RPE, so we added a chemical ML228 only on the apical surface of RP cells, which is ML228 is an iron chelator that leads to stabilization of HIF-1 alpha. So within 48 hours, we see HIF-1 alpha migrating to the nucleus and stabilize in cells. By 96 hours, more cells have it, and by 2 weeks, pretty much the entire monolayer has HIF-1 alpha in the nucleus, and you can also see degeneration of the monolayer with HIF-1 alpha overexpression. When we do this in 3D, we notice, so now we have three different tissues. On top is our control tissue. Remember, we're looking at it from the side view, so RP is like a flat line, and you can see the dense thickness of the capillary network. When we see from the side view, when we do the hypoxia, we can see capillaries proliferating underneath the RP. It's a typical type 1 CNV type hyperproliferation of capillaries, and we can suppress it all when we add anti-VEGF antibodies into this tissue. So this for me is a very good translation or clinical validation of this tissue, that we can recreate a well-known clinical phenotype and then use a clinically use a drug to actually suppress that phenotype in these cells. Very quickly, last slide, I want to show you that this model can also be used to study dry AMD between RP and choroid. Remember the complement model that I talked about in the middle of my talk? We use the same model here. We stress our tissue with activated complement. And now in this video, you'll see RP is in green, vessels in red, ApoE is in magenta. First of all, very similar to 2D, we see degeneration of the RP monolayer with complement stress. As I played a video, you can see ApoE deposits all over in between RPE and the capillaries, and capillaries have significantly degenerated in this tissue with as RP cells degenerate or as a complement attacks the tissue. You can see again very thinned out capillaries and complement deposits, or sorry, ApoE deposits, big ones or aggregates all around the tissue. From a side view, again, you can see RPE. Under normal condition, we see some complement, but significantly enhanced ApoE. We significantly enhanced ApoE in the presence of active complement in this tissue. So that's pretty much where we are now. We're kind of taking this further to make syngeneic tissue with different genetic backgrounds and see whether we can recreate these features and try to understand relationship between genetics of the cells and alternate complement pathway. So with that, there are a few people, I tried to mention most of the people who were directly involved. Several of the people whom I didn't mention are listed here, and some of the lab alumni are listed here. Well, thank you for your attention. And if there are any questions, I'll be happy to take it. Sorry, I ran a little over time.
Thank you very much. Very, very exciting progress. Anybody has a question, please drop me a note by chat. So I will start before people will start sending, hopefully, good questions. Only good questions are allowed. Let's go to Zach.
How are you, sir? Did you want to mention something before I go, or?
Well, my question is whether you look on production of the chromophore in the best culture you ever generated.
We have not looked at the chromophores just because we don't have the right techniques to do it. We have looked at the expression of all the genes involved in the visual cycle pathway, and we know they are there. But if you're interested in looking at the chromophore, we're happy to send you some cells.
Very well. Zach?
So kind of going off of that, I was wondering if you have an interest in like potentially incorporating immune cell types into your 3D kind of scaffolded structures of the RPE.
Yes, absolutely. Actually, that's an ongoing work. It was briefly presented at ARVO. We have tried to incorporate macrophages.
Yeah. I was wondering if you could model these kind of infiltrating pro-inflammatory or even anti-inflammatory monocytes or macrophages.
Yeah.
And also, have you tested basically if photoreceptor outer segment phagocytosis still occurs within these cultures?
So in 3D cultures?
Yes.
Yes, we have tested. It does happen in 3D cultures. We haven't done like an exhaustive comparison to 2D, but it does happen in 3D cultures. In terms of macrophages, we have been looking at.
Do you think that?
Go ahead. Sorry, go ahead. Zachary, we can't hear you.
Zach, we lost you. Let's move to a fellow named John D. I don't know who is that, but.
Oh.
Hey, Kapil. Great talk again. So I'm curious about, after your first part of the talk, I kept thinking about what subtype of RP are we looking at in all the different conditions? And so I guess I wonder, can you give us a little more insight about the P1 versus the P3, and are you seeing significant functional differences, or is it just a size difference? And how should we be thinking about the importance of the RPE subtypes?
So yes, I'm sorry I didn't clarify. So currently or previously we have only been making what we call P1, P2 subtype. So for the AMD assays that you saw, it was P1, P2 that was used. We are in the process of looking at or comparing functional differences between P1, P3, P2, and P3 that we can now make in the lab, and also looking at whether they're differential disease sensitive. So at this point, I don't have a good answer to your question, what are the differences, but that's kind of ongoing work in the lab.
Thanks.
Okay, let's move to Wahil Yu.
All right, so impressive talk. So my question is related to a previous question. So in your screening, you identify some molecule that can regulate the size of RPE. I believe you have done some functional annotation. What kind of do you want to share? What kind of pathway that's regulated the size of RPE?
Yeah, so this is still kind of ongoing work. We are kind of validating many of these compounds that we have identified and seeing which pathway is primary or secondary, because we think there are multiple pathways involved in each step, and some are upstream and downstream of it. So at this point, I don't have a good answer again for that, but this is ongoing work in the lab.
Yeah, so in your second part or third part of talk, you you do this. You have showed a staining of RP size during this competent condition, and also the hypoxia condition. I see briefly see the size of the RP kind of more become more inhomogeneous. So do you think that is somehow related to the compounds?
No, that's an interesting point. So no, what we see under complement activation or hypoxia condition, that is a completely different phenomena. There we see RP cells are degenerating and dying, and then the remaining cells are expanding to fill up that space. Has been shown in many other conditions, genetic conditions as well. What we do developmentally is a more stable feature of these cells. So once they attain that size, they stay like that. Whereas in, for instance, active complement, as we add the complement within 48 hours, we see cells are changing their size because the cells are dying and actually also undergoing dedifferentiation.
Thank you.
John Paul.
Kapil, thank you. So quick question for you regarding the light cycle and the activity of the RPE cell. So we understand some about the chronobiology and the activation and how it might change the structure of melanosomes. Have you done anything where you're simulating? I know the photoreceptors are not inserted into this model yet, but still, are you thinking down the lines about what the light cycle might mean? Because that we understand that these cells have a variation according to the time of day and the exposure to light.
Yeah, excellent point. That's one of the thoughts I have recently, and I've been trying to convince some people in the lab to start that project, and they go, no, we can't do one more project. You have to find somebody else. But no, you're absolutely right. So it's not easy to replicate circadian rhythm in cells, but I think by adding, one would have to, first of all, make a chamber that would be where you can control light even within the incubator, and changing the media would have to be also done at right times of the day, but then also add certain chemicals into cells at different time points to induce circadian rhythm. And that's a great point. One of the thoughts I had, but I haven't done anything on this yet.
Okay, thank you.
Maciej.
Hi, Kapil. Thank you very much for beautiful talk. I learned a lot. So I'm more interested in the properties of RP cells, like optical properties and morphology of RP cells. And you were mentioning about these processes that they can have this petal and finger-like character. Is there in this gradation of different cells, do you observe also the change in the density of processes, or the change, like smooth change of the petal to finger-like?
So we see a change in ratio of petal to finger-like depending on what population you're talking about. How smooth that is, I don't know, but it kind of correlates. Again, it seems always to correlate with the density of what kind of photoreceptors those cells are going to see. In areas where they would see both rods and cones, we see cells with mixed apical processes. Yes.
Thank you.
Kathy?
Wow, that was fabulous, Kapil. So I had two questions, actually relating a little bit to what he just asked. In your 3D cultures with the insults, with the hypoxia or your activated complement, do you see at the from beginning to end, do they transition not only in size from P1 to P2s, but do you see a transition in the apical processes from finger-like to the petal? I mean, sorry, the other way around, petal to finger-like. That was my first one.
Yeah, so we see more degeneration of the cells rather than transition to a different developmental subpopulation. So they are definitely getting bigger in size, but that is more like the cells are undergoing dedifferentiation. We see significantly increased upregulation of dedifferentiation markers. Essentially how we would see at the borders of a GA lesion, we see something very similar phenotype happening in the cells. Apical processes, essentially they start to stunt. So they get shorter, fewer and fewer, and with time, if you look at them with different days with the complement activation, the apical processes are really getting shorter and shorter, so they're losing that activity of polarization feature as well.
So have you started to try to rescue these phenotypes in the dish? So like using, something to enhance autophagy or any of the litany of things that you might do?
Yes, we actually have done that, but I took those 2 slides out. Maybe Chris would allow me to reshare my screen. I can show you 2 extra slides. If I still have them, let's see. Yes, okay.
But before you find this, I have a question to my friend, Dr. Friesler. Who has a nicer RPE behind, Dr. Williams or Dr. Bharti?
Okay, I'm not going there. Okay.
Smart man.
Okay, so do you see my screen again?
Yes.
Yes.
Yeah, so what we did was, using that assay that we had, we ran a small drug screen in the lab, and this was using a commercially available library called LOPAC, Library of Pharmaceutically Active Compound. So that means all of these compounds have some stage being used in some clinical setting. Not all of them are approved drugs, some are. So there are about 1,380 drugs in this library. And what it shows you, so we had 4 384 well plates. So each plate has different color, red, blue, green, and yellow. And we're looking for initially just cell survival here, right? So, and there were several drugs that popped up in the initial assay, about 40 or so, but most of those turned out to be false positive. And only 3 of them retained signal in the follow-up screens. And 2 of them have significantly reduced both ApoE deposits and increased the transepithelial resistance of RP cells. So that means we're not only getting rid of all the junk they're making, we're also improving their health. Which is coming first, I don't know. But there are 2 drugs. So one is called ACA, aminocaproic acid, which is a serine protease inhibitor and has been clinical use for many years. So now you can imagine what serine protease is. It would inhibit to block probably degradation of or cleavage of C3 and C5 into C3 and C5A. And the second one, L745870, is very interesting to me because this drug is actually a dopamine receptor inhibitor. And this can also actually connect to John's question because dopamine is a known circadian regulator. Both dopamine receptor and complement receptors are G protein coupled receptors. So we think, so when we looked at the activity of these drugs, this drug literally inhibited everything that the complement receptors activated inside the cell. The upregulation of AKT and ERK was downregulated, cyclic AMP was upregulated. All of that, this was doing wrong, this was doing right. So that now, so we're following this further, we're interested in this actually happens to be a Merck drug, and we're trying to convince Merck to allow us to use this. We'll see where that goes. But apart from that, it opens very interesting basic biology question, kind of the interaction between what the dopamine pathway may have to do with AMD or what the complement pathway may have to do with RP physiology, right? Because all of this suggests that there's low level alternate complement activation always happening on the apical surface of RP. What does that low level, and RP has apical C3, C5 receptors, does that have any role in RP cells? I don't know, but I think that's something we're interested in, may look at. Did I answer your question?
Oh, absolutely. Fabulous. Thank you.
All right, we have a lot of wonderful discussion, and for the truth and final comments, we're going to David S. Williams.
Well done, Kapil. That was really nice. I'm not sure what I'm doing here with final comments. I have a question about clarification, though. So you say P1 is from 0 to 1.5 millimeters. Is that radius, diameter?
That would be radius. So it's in diameter, it's about 3.5.
So that's sort of a bit beyond the fovea, but not the full anatomical macula.
That is correct. So what we think this is fovea and the perifovea, and not
Okay.
parafovea. That is correct.
Not the perifovea, right? Yeah.
Fovea, perifovea. No, fovea, parafovea, perifovea.
Yeah. So this leads. So the perifovea. I mean, you're saying that these sheet-like structures, processes, so to speak, which have been shown around cones a lot. I mean, there was a study from Steve Fisher's lab on specifically showing them on cat cones. You sort of go down towards them. But in the perifovea, which is beyond your P1, you've got as high a density of rods as you have cones right in the fovea. So so I'm trying to figure out whether you're saying these sheet structures are for cones or are they for high photoreceptor density?
I think they are for cones. So when we see cells that would be talking to both rods and cones, we would see both type of apical processes. And intriguing is that, and I don't have a lot of evidence supporting this, but we think that the density of, or the number of what kind you have, sheet kind or the finger kind, is directly proportional to how many rods or cones are up there.
So when you go then to the perifovea from the cone rich to the rod rich, and you're not changing photoreceptor density at all, you would expect to see a very sharp transition then in the anatomical, as you go out in the anatomical macula.
So that is true. And in fact, in vitro, that is what we see. When we make these cells, we see those cells have more of the finger-like processes, and less, and same cell has both of them now, right? They have finger and pedal, but they have more of one kind or the other kind. But what I showed you from P1, comparison of P1 to what we call P4, there we have pure one or the other kind. Every cell has only these, every cell has only finger-like. So yes, absolutely.
Okay.
This is really, a lot of this is ongoing, so we're still kind of digging and trying to understand more and more of this, and all of these questions are really valuable, and to get more into these details.
It's beautiful work, Kapil. Well done.
Thank you.
Well, I forgot about one person, Deepak. Sorry, so Dr. Williams will not be the final commenter on your work, but we go to Deepak and he will conclude this session today.
So I was just curious if you would expect a difference in the growth of cells across the scaffold in dark adapted versus light adapted conditions.
Well, maybe not growth of cells, maybe even growth of apical processes may be different under different light conditions. It's known in many animals, like I think frogs and other species, it's known that apical processes under light conditions grow all the way up and then they retreat under dark conditions. Maybe, I don't know if the human cells do that. This is going back to the question what John was, I think, asking too. We have to make those conditions work in vitro, first of all, because I'm not convinced whether we can nicely control light and dark under in vitro. I think we have to do it chemically on top of controlling light to these cells, because I don't think these cells are light sensitive per se. We have to first add the right chemicals to activate the circadian pathway to make them light sensitive, and then we can do this experiment. It's an interesting thought. I don't think it's known that human RP cells regulate their apical processes by light, but we have a way of looking at it now.
But do these cells always develop in light, or is there a light-dark cycle?
Well, they develop in the incubator, right? And
which is, I don't know. I mean,
so incubator, when it's
culture.
closed, it's dark, but when it's open, it's light. And
okay.
The same incubator has hundreds of plates and media is changed throughout the day. I would not be convinced that there's any cycle in these cells.
Yeah. Thank you.
I like to do a much more controlled experiment here.
Thank you, everybody. Have a wonderful weekend. And I will see you with Dr. Campuchar a week from now. Hopefully he will not be grumpy.
Thank you, everyone.
I don't know.
Yes.